All right, please join me in thanking Patrick once more. Okay, next up is our very own Matt Perich. Matt is professor at the University of Montreal and an academic member at MILA. He has a lab at the neuroscience department here, and he's been doing very high-level work both in sort of computational neuroscience and also in experimental non-human primate stuff. And so today, Matt's going to talk to us about common neural basis for behavior across individuals and species. So please take it away.
Thanks for the introduction. Thanks for the invitation, Kareem and everyone else who organized to speak. So, when I realized I was asked to speak in a foundation model section with Blake Richards, Patrick Minot, and Eva Dyer, I realized I had nothing to contribute on the AI side over these brilliant minds. So I'm going to put on my neurophysiologist hat and talk about some much more basic, fundamental, science-oriented results from my lab about why I am sharing the cautious optimism of people like Patrick Minot about the idea of neurofoundation models. Maybe trying to convince you that it's not just data trickery that we start to do things like transfer decoders from a monkey to a human, that there's actually maybe something in us as species and as evolutionarily evolved beings that makes these things feasible. So with that introduction, what I hope by the end is that you can be convinced that neurodynamics are this idea of temporal evolution of population activity in the brain, that this is something that for specific actions is conserved across individuals, and that it's actually capturing something quite fundamental about the functions of the brain or maybe the computations that a circuit performs, and that these are inherited and preserved across different species, and this is essentially where we put in our step 6 and profit with foundation models. This is what ChatGPT 4.0 thinks the foundation model is, so I think that basically summarizes Patrick's talk in one compact little image, so that's great. All right, so to sort of set up the talk, different individuals, like for example, 2 macaque monkeys, they'll do very similar behaviors, and they'll do them in very similar ways. You put 2 macaques in the jungle, they're both gonna reach for the mangoes. It's not that one's gonna reach like this and the other is gonna reach like this. There's very species-typical ways in which we control our bodies. And I would argue that this is actually evolutionarily imposed. I mean, the brain of the macaque evolved with the effector of the arm. This would also be true of a fish that whips its tail or its fins, a mouse that forages for food in the forest. And besides the evolutionary kind of development of these things, there's also this sort of developmental development to these things where as you go from a newborn baby or a pup up to an adult, you sort of have the expression of these genetic codes, which gives you the brains that let us do other things that we do, essentially. So then one of the ways in which theoretically you might conceptualize this is that whenever the monkey goes and reaches for the mango, there's some sort of landscape of possible activity states that could be taken in the brain. And whenever he makes this movement, activity traces out some pattern on this quote-unquote landscape. And then if we reconceptualize sort of development as an instantiation of that landscape for different individuals, we can make the hypothesis that there should be very similar dynamics of how the brain activity evolves during these behaviors. Okay, so how do we test this hypothesis? I'll give you a crash course on neuromanifolds, which is my favorite term to use, so I have to put it in every talk. And it's based on this idea that if you have some population of neurons, here it's some number of neurons in the brain, each circle is a little cell, say in the motor cortex, which is a focus of this talk. And we can put an electrode in and we get some activity. Intracortical electrophysiology would give you action potentials as one of the most relevant signals. But the important thing is that as much time as we spend trying to understand these patterns of like spike trains, really this is the output of the cell. And if you're doing some black box style computation or like reverse engineering, like you don't care as much about the outputs as the relationship between the inputs and the outputs. Now, of course, that's extremely hard to get access to experimentally, so we have to try to infer things through analysis. And this is where I think the power of population analysis is, where we just completely acknowledge that all these cells don't live in isolation, they're all connected, they all influence each other. And depending on these connectivity profiles, you can have these certain modes of operation. So here colored by green or blue types of colors, where if you ping one cell, you're gonna propagate through this network with some pattern. Ping a different cell, you'll get a different pattern. And these are what I would call these neural modes, and we can actually estimate those by doing population recordings, we would argue. So in this cartoon example, we can put 3 electrodes in the brain, and then define a state space where each axis is the firing rate of one of these cells. And then over time, if you ask, say, a monkey to reach for a mango, you'll trace out some trajectory through this space. And what we find empirically is that in this full possible space of interneuronal firing rates, you don't actually explore all of the possible states. They tend to be confined to some lower dimensional type of plane, which we would call then, in jargon, the neural manifold. So I've written lots of papers, and many other people have written lots of other papers kind of about these ideas. Actually just had an article come out in this neuroscience magazine, The Transmitter, which I specifically try to address a lot of people's complaints about the idea. So if you're not convinced, maybe that article will at least be interesting to you. And also one of the complaints is that people say like, well, obviously, why would there be a hyperplane in the brain? It doesn't make any sense. And I agree, it doesn't make any sense. And there's lots of evidence that although we like to think about these things schematically as planes because we can draw them in 2 to 3D, in fact, the actual types of manifold-like structure we see in the brain is much more complex, from Adrian's work in the head direction cells, where they show there's these ring attractors. There's toroidal hippocampal manifolds. Worms, whenever they locomote, have this kind of weird shape that is consistent across individuals. What's really important here is that regardless of whether it's a plane or regardless of whether it's a sphere or whatever this surface is, whenever the animal is producing some behavior, it's tracing a trajectory along this manifold, and that's what we're thinking of as the neurodynamics. So these are just constraints in the network that are giving you meaningful structure in the temporal evolution of population activity. And for the rest of this talk, I want to just take that as the framework and then start to characterize aspects of that population activity across different individuals and species. So this is work with a good colleague of mine in London, Juan Gallego, led by postdoc Mustafa and PhD student Joanna. And here we set out to directly test the hypothesis from the beginning of the talk, which is that if you just have 2 monkeys do the same behavior, do they kind of do it with the same neural implementation? So how do we do this? We think back to our landscape idea, which we now can formalize as something of a mother manifold, let's say. If we put 3 electrodes in 2 different monkeys and get 2 different sets of cells, there's going to be some idiosyncratic structure to the trajectories that we observed in those 2 monkeys. But if we think of the true underlying bits and bobs about it as inherited from some maybe genetically constrained master construct, then it's really just a matter of alignment. We have a space of 3 non-overlapping neurons from non-overlapping brains. We just have to find, in theory, transformations that would align these, and then we see that they're actually quite similar. So we test this using some experiments that I did in monkeys. So this is a very simple movement task where monkeys are cued to move in 8 different reach directions. So you get trajectories like this. We record for the motor cortex and study the motor system. Motor system is nice because you can see what the monkey does, so you get a very vertical and easily interpretable output to relate your activity to. I think that these types of principles should be true in a lot of other systems, and we could talk about that offline. I don't have time to go into everything we're doing along these lines. So on the left is just one example monkey for 8 different reach directions, and then the other 2 plots are of 2 other monkeys, and you see pretty clearly these monkeys are good at this, and they all do it the same way. Obviously, they were trained to, makes complete sense. So then if we go in and we put some electrodes in the brain, and here we can record about 100 neurons of activity from the motor cortex for each of these 8 reach directions here in the different columns. And then you see the heat map where brighter colors of more active neurons, and then the bottom is kind of the position of the hand during these movements. So for one monkey and a second monkey, you can see this behavioral position, the purple at the bottom, they're extremely similar. Obviously the neurons, they're different brains, there's no correspondence. So then we want to use some of these population techniques to ask okay, how similar is the underlying structure of these seemingly random different types of signals? So the technique we use is based on dimensionality reduction. You can do it a lot of different ways. The very simplest way, which we like, is just principal component analysis. So run a PCA, take 10, 20 dimensions, depends on your data set. Here we can visualize as the top 3, where each color is a different reach direction. So you'll see this sort of structured organization by target location based on these colors. For the 2 monkeys, there's some similarities, maybe there's some differences. And then again, the simplest possible thing you can do, canonical correlation analysis, which is a linear alignment, and just say, okay, if we just assume these things are linear systems, and we just need to linearly align them, how close do we get? And it turns out pretty close. So this is the same data in a new aligned space that maximizes the correlations between these signals. And then by this method, you kind of get this correlation readout, which is necessarily monotonically decreasing rank ordered as you do dimension by dimension alignment. So as a reference, we kind of give in gray if you just do sets of trials within a monkey doing the same task, and then in maroon, or the kind of reddish maroon, if you do it across these two monkeys. And then a shuffle control at the bottom is a lower bound. What you see is basically the amounts of correlation between these trajectories in this task is really at the ceiling provided by the lower within monkey correlation. So from the extent of these linear alignment techniques, like monkey A looks no different from monkey B, could be any monkey. So this is nice because it shows that there's some sort of generalization. And if we then kind of think about what's happening, I mean, the task itself is a constraint. Both monkeys do the same task, they've been trained to do the same task, but it's a two-dimensional task. You can essentially explain all of the kinematic variance with 2 dimensions. So you sort of get 100% alignment of these top 2 dimensions for free in some sense. What you don't get for free is everything else here, where you're so much better than chance. So there's something intrinsic to how these dynamics are generated that seems to be preserved. And if we just do a bunch of sessions with a bunch of monkeys, there are about 6 monkeys in this paper, we see that this is pretty consistent. And I want to point out that these linear alignments are, even theoretically, kind of make sense. So this kind of stems from some work I did with Sarah Soya in my PhD, where we were inspired by Sergei Ganguly at Stanford and his random projection theory ideas for neuroscience, where if you think about every electrode you're putting in or every neuron you're recording as some random projection of some true underlying process, it actually should be linearly reconstructible, even theoretically, to be able to align these dynamics, provided you have enough samples to estimate these things through these linear processes. So it all fits. Now, is this meaningful? One of the ways we can test this is just through decoding. So we just take the brain activity, put it through an LSTM, and then predict the 2D kinematics of the monkey's hand. And here's an example of what this looks like. So what we actually want to do is predict these kinematics, but see if we can use another monkey's brain activity to predict a different monkey. So the black here would be the actual kinematics, and then if you sort of train a decoder on that monkey and test on that monkey, you get the gray trace, so kind of a within performance. And then if you just take another monkey's activity and play it through that decoder, you get the green trace. So it's clearly missing all these dynamics. Whereas then if you take this aligned activity and play it through the decoder, now you get the blue trace, which is essentially across all these different reach directions, just as good as our within monkey performance, which we can then quantify using kind of a decoding accuracy. So essentially, across monkey alignment does almost at the upper bound of within monkey performance just by this simple rotation of neurodynamics. Okay, so it's not just that we have high dimensional statistics and we can align them any way we want. They actually map onto behavior in a really meaningful way, so that's great. And I'll step you through a couple controls that we do, just for the sake of time. I'll go quickly. So one of the things you could argue is that, okay, both monkeys are also not just moving, they're getting the same sensory feedback. Could it just be trivial that you get these sort of like rotational loops for different reach directions that are just some sort of like self-predictive feed-forward system, and is it actually some kind of computation? So we do a test where we ask monkeys not to move, but to just think about moving. So there's no sensory feedback, and then we do the exact same across monkey alignment, and we get the exact same result. So even when the dynamics are, in a sense, like internally generated, we can still see these alignable features across monkeys, which is great. And also, is this some primate motor cortex thing? We partnered with Josh Dudman and his postdoc Joan-Chul Park at Janelia Research Campus to test the same thing in mice. So here they have a task where the mice reach out and pull on a lever. They record from the motor cortex and the dorsolateral striatum, which is an evolutionarily older structure. I'm just going to show you the motor cortex here. The striatum looks essentially the same. So you can see that the alignment does work. We're above the control, but you can also see it's not as high as the kind of within mouse. So then we ask, well, why is that? Okay, is a mouse different than a monkey? And we actually think it's not. So if you look at the correlation of behavior, which is on the horizontal axis here, it's actually lower than the monkeys, almost completely linearly explaining the difference in this neuroalignment as well. So it's not that mouse brains are different, it's that mice are just not very well trained at this task. And that's limiting our ability to see what's kind of common. So it's really emphasizing this is a neuroalignment, it's about neuroimplementation, but similar behavior is extremely necessary to see these types of effects. Now, for the sake of time, I will just show you kind of half of this slide. You could also say there's a lot of structure in the task, and I like to think the planning activity got away from this somehow, but we wanted to ask, is it really necessary to have a structured task? So we have these monkeys do a whole bunch of random movements in the workspace, and then Joanna, the PhD student, did this heroic alignment algorithm where she could pick different starting and end points of the same duration and same speed, and just build this library of different matched movements between the monkeys. So you can get something like this, closer to 30 matched conditions instead of the 8. And what we find is that we get exactly the same performance. And in fact, once you've seen about 9 of these, if we randomly select, you'll get saturating alignment performance as you add more conditions. So once you've seen enough to understand what it means to move your arm in 2 dimensions, then just giving it more complexity doesn't change your alignment at all. So you're really capturing something, we think, fundamental. And then one last thought on this across monkey comparisons is what we've been showing with this canonical correlation analysis is a geometric alignment of these signals. But really, these underlying dynamics can be quite powerful and can be quite different in these cases. So this is an example from a paper from Valeria Monte's lab, where you can see this sort of dynamical system flow fields that are guiding how these trajectories are evolving in this 2D space. And although that the exact trajectories we're seeing in black are the same between the left and the right panel, one is actually generated by a line attractor and one is generated by a point attractor. So you can have geometrically identical activity with different underlying mechanism or computation. So fortunately, some really cool work from Ilafit's group from NeurIPS last year is designed essentially to disambiguate this case. So I won't get into the math. Guillaume would know it better than me anyway. But the idea is to do some sort of linearized transformation into a linearized manifold in order to look at the actual temporal evolution of these trajectories. And then once you're in this new linearized space, you can do Procruste's alignment between 2 different sets of trajectories. And it's not about their shape now, it's really about their time point by time point evolution. So based on that alignment in this new transformed space, you can actually make inference about similarity of dynamical systems, not just these geometric shape. So thanks to the fantastic postdoc Olivier Caudot, we have tested this on these monkeys. So we took this random movement data, which gives us a nice complete sampling of this manifold space, and then tested this alignment. And if you just look at these kind of values in this aligned space with the Procruste's alignment, so here instead of like a similarity like correlation, you get a dissimilarity, so now low values are actually more similar. And we see that by the way that this is plotted on these arbitrary scales, this looks quite low. We're below 0.1, but what does 0.1 mean? Who knows? It's an arbitrary space, so we need a reference. And the reference we choose here is to actually look between 2 different behavioral contexts. So if you take these monkeys and say, when you're planning a movement versus whenever you're actually executing that movement, this is what we get. So within a monkey, 2 different actions from the same neurons in different contexts gives you much more dynamic dissimilarity than the same action across monkeys. So I think it's not just that whenever you have some movement in space, you get some shape of a trajectory, there's actually these underlying mechanisms that are actually quite preserved. All right, so for the rest of the talk, I'm going to try to go quickly through another couple pieces of story. So one is to say that we think that there's something in the evolution that is giving it this common structure. And this is expressed during development in a way which is explaining why we get this common mechanism across individuals. And one of the ways in which you could think about this is sort of this like manifold being shaped during development. So it's a really hard thing to tackle experimentally. I mean, people certainly can do recordings throughout development, but I wouldn't necessarily say that's an easy task. So one of the ways we can get at this is, if you want to understand, how your kind of like initial baby babbling to try to grab the ball turns into your ability to like roll the ball whenever you're 3 or 4 years old, is to study this in simulated systems. So this is again some work with Olivier Caudot and Guillaume Lechois, who's here, as well as Nanda Krishna, who helped on some things. And the idea is to understand, okay, if there are multiple ways in which a network could develop in order to produce some behavioral output, there's a few different theories you can compete. One of them is to imagine, okay, the monkey wants to grab the mango. He's too young to know how to grab the mango. So he tries to grab the mango, and time point by time point, his sensory systems tell him, you're not going to the mango, and then he adjusts, and then he adjusts, and then he can grab the mango. And over time, he could just kind of learn to do that more efficiently. So this is a highly supervised process where your loss is based directly on your error relative to this mango target. We can think about it differently in more of a reinforcement learning paradigm where you have a mango that you want to get to, so you try to get to it. you realize you failed, and you try to figure out why you failed, and on the next try you get better, and you go through this iterative process. And these can both explain a lot of the behavioral data, but they make very different predictions about the learning trajectories, and we would like to think about mechanism and implementation. So we wanted to test if either of these better explained how kind of a monkey brain came to be. And what we did was set up a simulated paradigm where we have a biomechanical model of a monkey arm, so 2 links driven by 6 muscles. And then we have a recurrent neural network which actuates these muscles in order to move it in a 2D space, much like the monkey's arm did in the data I showed before. And then once we have this system, we can either train it through a direct supervised learning process where we just compute the error between where you are and where you want to be, or we can implement through PPO, kind of a very similar reinforcement learning process where you want to evaluate the success of your actions based on the states and the rewards you get. So what's great here is that the exact architecture of control is completely the same. All that changes is from de novo state to behavioral production is the teaching signal that this network was given. And what we see is that both of these networks can learn to do random movements through some training process. This is loss over training epochs. There's small differences, but they're not that appreciable. And then once you ask them to produce movements, you can see that both RL trained on top and supervised learning trained networks on the bottom can produce this kind of center out task. They weren't explicitly trained to do that task. They were trained from random movements, but now they produce this task when cued. And exactly, this looks a lot like the 2 monkeys that I showed before. This is the same data from the beginning of the talk. And then what we want to ask is, what is the kind of neural implementation? How does that differ between these networks? So if we just do PCA on the unit activations of an RL network and a supervised network, we immediately see visually there's qualitative differences in how these trajectories evolve. Just by eye, you might be saying, hey, that bottom monkey thing looks more like that RL thing, doesn't it? And we can actually quantify this using DSA. So this is the dynamical similarity analysis we showed before, where we just take the monkey neurons, compare them through the same exact method that you just saw to the unit activations. And as a reference, what we do is sort of shuffle time points, and that's these box plots you see at the top. So that's like our upper bound. How bad could you possibly be? And while we see that supervised learning, it doesn't match the monkey a little bit, the reinforcement learning models actually match the monkeys much better. So we're thinking now that reinforcement learning is a better kind of model or theoretical idea for how these monkey circuits developed than a supervised process. Now you might think, if we're trying to make this some kind of developmental evolution story, there should be something about the effector that matters, right? Because we evolved to use our arms, that should be shaping the representations, and we can test this quite directly by just replacing that 2-link arm with a simple point mass driven by 4 pseudo muscles in 4 different directions. And when we make this change, we essentially see this effect go away. So now there's almost no difference between RL trained networks and supervised trained networks, and they're both much more dissimilar than controlling the 2-link arm. So it's not just that reinforcement learning is a nice model of how this brain might have developed, it actually requires you to have something that looks like a monkey in order to get something monkey-like in the brain. And I think this is really interesting because it matches my expectations for like babies, right? Babies babble and they flail their arms, and eventually you learn that you can grab your mother's hand if you really know what you're doing, and then this is kind of how we get better at motor tasks. Okay, so you can hear a lot more from this guy. This is what he'll look like next to his poster. And it's here at Maine, so go check it out. And we also put up a preprint a couple weeks ago, so yes, definitely interested to hear your thoughts. And in my last maybe 5 minutes, I want to tell you one more piece of story. So if we're thinking of this potentially common across individual dynamics as being of developmental origin, that would then say, well, there must be some evolutionary basis. So there should be some basically way, like, can we test this using actual models of neurodevelopment? So in some normal way in which this forms, you get 2 monkeys that produce the same things, but what if you have, say, a neurodegenerative disorder or like a transgenic defect? So you would get some sort of divergent developmental trajectory. And I don't have time to tell you these results, but we're actually testing this in collaboration with Adam Hentman's lab at UNC. Reza, I don't know if he's here, he also has a poster here at Maine where we can talk about the results. Same types of analyses you just saw, but now comparing across different types of mice that have different genetic manipulations and showing how we can and can't kind of change what you see across these individuals based on their developmental background. And then one more little extra eye candy for the end of this talk is, now can we really try to make some sort of evolutionary origin more directly? I'm going to channel my inner Paul Zizek. I don't know if he's here. This is a great figure from one of his papers where you can see how we sort of get this elaboration of different species over millions and millions of years. And we'll kind of zoom in on this little piece at the end, which is whenever the rodents and the primates sort of separated about 80 million years ago, 100 million years ago. And we can kind of make the prediction, okay, if two monkeys, when they do the same thing, they have the same sort of implementation, it's really not that long ago that we might have diverged from our rodent. And in fact, I've been studying the motor cortex. Mice have a motor cortex, monkeys have a motor cortex, humans have a motor cortex. We can make the hypothesis that actually if they inherited from the same region, they should also have the same implementations, so we should also be able to see the same principles in a mouse and a monkey. So we can test this directly, as long as we have a mouse and a monkey doing the same behavior. Fortunately, we do. So on the right is a task that I've trained a monkey to do in recorded motor cortex as they pull on these robots. On the left is kind of the data I showed before of a rat or mouse reaching out and grabbing things. So we have some quite similar behavior across these species. In both, we can get motor cortical spiking recordings, on the left with neuropixels, on the right with the Utah array. And then we can do this exact same test we've done before, where we take trial behavior matched alignment of monkey and mouse, and then do dynamical similarity analysis between the populations that we get. So what we're seeing here, and again, remember, lower values are more similar. In the red is if you just take two mice and run DSA. In the blue is if you take two monkeys and run DSA in this task. Mice are lower, maybe there's something profound there, maybe there's not. There's a lot we could talk about. I think what's more interesting to me is if you take these monkeys and put another electrode in the same brain, but just record from the somatosensory cortex. The somatosensory cortex is deeply connected to the motor cortex. I would actually think of them as the same dynamical system. We'll actually get a little bit of an increase in, or decrease in similarity, increase in dissimilarity, but it's sort of roughly the same. That provides us another kind of reference for how reproducible these results are. And now if we run within the motor cortex across these species, we're getting essentially the same values as across monkeys. So the limit of dissimilarity you can see between a mouse and a monkey is actually set by the across individual dissimilarity in these NHPs. And the same reference we used before, if we just take the same monkey and change behavior or context, we get a much higher thing. So it actually seems to be true that, like, almost somewhat mechanistically, that a mouse really is similar to a monkey in terms of how the motor cortex might be driving their arms for, like, reaching and grasping tasks. So then I think that's where we kind of get back to our summary of trying to show that we see things across individuals, they seem to capture real processes, maybe even developmental, and now they're also there across species. So I don't think it's much of a leap to say that if you're seeing all these things across species that almost seem to be mechanistically preserved, that a foundation model is such a crazy idea. Like, why should we not be able to, with enough of algorithmic sophistication, extract all this information unsupervised and use it for lots and lots of useful tasks? And that's my sales pitch. So I want to thank everyone who's helped, especially the work that I talked about today, like Olivier and Guillaume, Juan, Mustafa, Joanna, and then the other poster, Reza, Adam, and Stefan. I have many other collaborators, so sorry if you were not mentioned. And then I'll take some questions.
All right, thank you, Matt. We'll have, in the interest of time, we'll take maybe one or two really quick questions. I think Chad was the first one on the side.
Hi. So are there lessons to be taken from reinforcement learning that could help us optimize training for a brain-computer interface? Like in terms of reward and, I don't know, like delays, things like that?
That's a great question. I don't know if I'm going to think of an intelligent enough answer in the 1 minute I've been allotted for this Q&A.
Okay.
But I would actually love to brainstorm offline, and you probably have better ideas than I do.
All right. No, that's good, thanks.
Yeah.
Is there time or?
Yeah, please. Yeah.
Hi, Matt. I loved your talk like I did last week in the first talk. So I'm thinking a little bit about what you presented in terms of the cross-species similarity. And you're using a motor task, which makes a lot of sense to me, but I'm really interested in higher cognitive processes. And we know that if we wanted to look at the same kind of mechanism or process, we may need to use different tasks across species. Would you be able to see the same kind of data in being able to, this kind of similarity between species if you're using different tasks that tap into the same process?
I can conjecture. I think yes. I think the most interesting question, though, is when do you not? And I think there has to be a point where you don't. I mean, there are things a mouse can and will do that a human can never do. I mean, there olfactory sensitivity, for example. Like, there's going to be certain tasks, certain parts of the brain, certain things where there can never be an alignment beyond the most trivial levels. This is also where I get excited about the idea of the foundation models, because once you're sort of at a scale where you can digest a lot of data from many tasks, from many individuals, from many species, if you have the right cleverness built into, say, how you embed the data, you can actually use that to tell me which samples do you think are actually similar between these species and which samples do you think are dissimilar. And then that becomes an avenue for hypothesis generation and testing in some ways. So we can actually go find out, well, when do we expect these things not to transfer? There's one tangible comment I can make there is there's a great paper from Dave Freeman's lab at U Chicago from a few years ago where they show that in parietal cortex that the training history of how you build up a multi-contextual cue task for two different monkeys changes the actual dynamics you see in these brains. So for a task where you have to integrate multiple information streams at different orders, if you've learned to do it one way, you actually might generate different solution dynamics than if you learned another way. So that's saying that once you get out of the motor system where you're intimately connected to quite vertical like effectors and plants that are very common across individuals and even species, then maybe it's not, it's much more difficult, or at least you have to be much more considered in how and when you find similarity. I would still think of that as the same concept though. It's like behavior broadly defined. If behavior is similar, I would think it to be similar dynamics. Some behaviors can never be done into individuals, so they can have no similarity. Some, they may be so complex that it's hard for us as external observers to understand whether they're actually doing the same behavior or not. But whenever you can be confident they're matched, I would expect it to almost, if not always, transfer.
Okay. Please join me in thanking Matt once more.